Good day, my dear students. You are welcome to class of BCH 407. Today we shall consider topics 4 and 5, or topic 4 and topic 5. Starting with topic 4. The title of topic 4 is Nature of Drugs and Transport Across Membranes. Nature of drugs and transport across membranes. Now, the objectives for topic 4 are just 4. First, we want to define drug and their nature. Secondly, we want to understand the structure and permeability of biological membranes. 4, 3, I beg your pardon, we should be able to distinguish between active and passive transport systems. 4, at the end of the lecture, we should be able to explain drug passage of membranes and the factor that limit such passages. Drugs and their natures. In our previous class, I defined drugs as any substance intentionally taken with the aim of achieving a therapeutic outcome. Any substance intentionally taken with the aim of achieving a desired therapeutic outcome. Of course, drug is a matter, that is, it has weight and occupies space, and therefore, like every other matter, exists in 3 forms: solid, liquid, and gas. Drugs have properties. Their general properties refers to the features that defines its identity, behavior, and effectiveness in the body. These properties can broadly be categorized as structural, physicochemical, biochemical, pharmacokinetic, and toxicity characteristics. The table before you display the summary of major drug properties. By your left, you have the properties, and your right with the details. The table comprises of structure, physicochemical, physical, chemical, pharmacokinetic, and toxicological. Now, this table or the structure before you, or the table before you, is showing you the 3 drugs with their general properties. We have paracetamol, that is acetaminophen, and aspirin, acetyl salicylic acid, and metformin. You have the chemical structure, molecular formula, molecular weight, mechanism of action, and therapeutic uses. Let's look at the structure and permeability of biological membranes. Biological membrane is a thin layer that forms the outside boundary of a living cell. Membranes consist largely of a lipid bilayer, which is a double layer of phospholipid, cholesterol, and glycolipid molecules that contains chains of fatty acids. Now, this slide is presented to give you an idea of the membrane structure. The first and the second, depending on which one you choose, are all examples of membrane. The first, by your left, is a very simple structure showing you the component of the membrane. And of course, to your right is also another structure of the membrane showing you its complexity, containing the integral and surface proteins. What are membrane functions? The membrane or the biological membrane function to keep toxic substances out of the cell. They allow specific molecules to pass in between the cell and outside environment. This is due to the fact that they contain receptors and channels. The membrane also separates vital but incompatible metabolic processes conducted within organelles. Now to the crux of the matter, transport across membrane. The medium that facilitates movement of solute in and out of the cell is called a transporter. These transporters are proteins, as shown earlier in those uh, pictures um, describing the biological membranes. There are two classes of membrane transport proteins, and these are known as pumps and channels. Another word that can be used for pumps are carriers. Membranes regulates the passage of solute through biological membranes. This is because they are semi-permeable. Membrane transport refers to how substances move across biological membranes. Next is What are the mechanisms of transport across membranes? There are three types of mechanism, namely passive, active, and bulk transportation. Among these three, passive and active transport constitute the main type. Passive transport requires no energy. It is a process where substances move down their concentration gradients. There are three types of passive transport systems. The first is simple diffusion, where small nonpolar molecules like oxygen and carbon dioxide go through the membrane easily. The second is facilitated diffusion, which means the diffusion is facilitated. That is, the molecule to move into the membrane is helped by a membrane, another membrane proteins. That is why it is referred to as facilitated transport or transport. Osmosis is another means, but a very special case of diffusion. It is of note that water moves across membrane through special channels called aquaporins. Now, active transport mechanism. This type of transportation requires energy as against the passive movement. Substances involved in active transportation move against their concentration gradients. That is, they move from a position of lower concentration to higher concentration. And of course, to do that, it will require a little force, which is provided by energy in form of ATP. Now, let's look at the types of active transport. We have the primary active transport and the secondary active transport. In the first case, it involves direct use of ATP to pump substances into the cell. Example is the potassium or the sodium potassium pump or the sodium potassium exchanger. So anytime you hear sodium potassium pump or sodium potassium exchanger, it means the same thing. Secondary active transport. This utilizes energy from the movement of one substance down its gradient to move another substance against its gradient. Example can be seen in symport, antiport, and uniport movement of substances through the membrane. A quick look at the diagram before you will give you a clear understanding of what this term implies. The uniport system of transport, the antiport system of transport, and the symport system. In the case of the uniport, it's only a molecule that is moved into the cell. In the antipods, two molecules are involved, but in opposite direction. In symport, again, two molecules are involved, but moved to the same direction. The third mechanism is the bulk transport mechanism. In this case, we have endocytosis and exocytosis, which you are already familiar with. In endocytosis, cell brings substance in by engulfing them in a vesicle. In exocytosis, cell expel substances by fusing a vesicle with the membrane. Drug passage through the membrane. There are only two ways for a drug to get into a living cell. As discussed earlier, these ways are passive diffusion and active transport. There are factors that could limit these two processes, that is, of active transportation and passive transportation. And these factors are: 1. Drug molecular size. 2. Concentration gradient. 3. The membrane thickness. 4. Surface area of the membrane. 5. The pKa of the drug. And 6. The pH of the solution. These are all factors that can influence the intake of drug into biological membranes. Of course, let me hint you here that when we talk of intake of drug into biological membrane, we are actually talking of drug absorption into the cell. Now, topic 5. Drug receptors and distribution. Let's do a quick rundown on the objectives of this class. By the end of this lecture, you should be able to define and describe drug receptors and their types, explain drug receptor interaction and selectivity, understand the process and factors affecting drug distribution, and relate drug distribution to drug action and side effects. What are receptors and their functions? Receptors are molecules present either in the surface of cell or in the cytoplasm. They act as mediators of signals between or within cells. There are various types of receptors. Few of them are multi-subunit ion receptors or channels, the G protein coupled receptors, enzyme-linked receptors, and transcription factor receptors. We will look at each of these one mentioned in turn. Starting with multi-subunit ion receptors. These are ligand-gated ion channels. They are sizable, composed of 4 or 5 subunits. When these receptors are activated, they form a membrane spanning the ion channels that permit flow of ions such as sodium ion, potassium ion, calcium ion, or chloride ion across the cell membrane. Second on the list is the J protein coupled receptors. These are integral protein or integral membrane proteins characterized by extracellular amino terminus with 7 transmembrane alpha helical segments and intracellular carboxyl terminus. G proteins are capable of detecting a diverse array of signals, including photons, ions, proteins, neurotransmitters, and hormones. The diagram you see here is activation or activity of the G proteins. You can see more of this in the reference text. Number 3 is enzyme-linked receptors. They represent another primary class of cell surface receptors, first identified due to their involvement in mediating cellular responses to extracellular signaling proteins that stimulate growth, proliferation, differentiation, or survival in animal tissues. The diagram before you, or diagram shown before you, is that of enzyme-linked receptors. It has a ligand binding domain, the intracellular active enzyme domain, the transmembrane domain, as indicated in the picture before you. The fourth is transcription factor receptors. They are a group of proteins responsible for reading and interpreting genetic materials or blueprints encoded in DNA. They bind to specific DNA sequences to regulate gene expression, which is essential for numerous vital cellular processes. Now, this is a pictorial of transcription factor receptor. Let's take a look at drug receptor interactions. First of all, we may want to know what a ligand is. It includes drug, hormones, neurotransmitters, which can be specific and bind reversibly or irreversibly to receptors. We may say that there are things or anything that can bind reversibly or irreversibly to receptors. Binding can activate or inactivate receptors, leading to increased or decreased cellular activities. Affinity and efficacy of drugs. We are going to use the concept of lock and key to explain this phenomenon. Affinity is a measure of the strength of a binding interaction between drug and its receptor. I take it again. Affinity is a measure of the strength of binding interaction between a drug and its receptor. What does it indicate? We talk of high affinity and low affinity. High affinity means that drug molecule has a strong attraction for its receptor and will readily bind to it even at low concentration. Low affinity, on the other hand, implies that a drug molecule has a very weak attraction for its receptor and requires a higher concentration to achieve significant binding. Efficacy, on the other hand, describes the ability of a drug to bind and activate the receptor or its receptor and to elicit a biological response. Just as we mentioned for affinity, we can also have high drug affinity or low drug affinity. Just as we have high drug efficacy and low drug efficacy. For high drug efficacy, it is used to mean that a drug can bind its receptor and cause strong activation, leading to significant biological response. While low efficacy, on the other hand, implies that even when the drug binds to its receptor, it produces only a very small or submaximal response, even at a high concentrations, where all the receptors are occupied. These drugs are called partial agonists because they can still bind to the receptor but do not trigger same maximal response as a full agonist. We can still talk about zero efficacy, which implies that the drug binds to the receptor but does not cause any activation or biological response. These drugs are known as antagonists or blockers. They can prevent agonists from binding to and exerting their effects. Now let's look at drug distribution in the body. To understand this concept, all we need to do is to provide a definition for drug distribution. Drug distribution is simply a process by which drug is dispersed throughout the body's blood system, including tissues after absorption. Now, there are factors that could affect distribution of drugs. These include flow of blood to tissues, plasma protein binding, tissue permeability, defined by blood-brain barrier, placenta barrier, and what have you. Then the physicochemical properties of the drug, that is, its size and lipid solubility. Now, protein binding and free drug concepts. Only free drugs can interact with receptors and exert effects. If the drug is bound to in any way, it cannot interact freely with the receptor and therefore cannot exert effects. Protein binding acts as a reservoir, influencing duration of action and potential drug interaction. Remember that these receptors act as they are mediators for signals or cellular signals. Now, summary of lectures. I'm summarizing lectures 4 and 5 simultaneously. Lecture 4, we talked about drug and membranes. Lecture 5, we talked about receptors and its distribution. Let's look at lecture 4. We talked about drug properties by way of structure, membrane structure or the lipid bilayer, and then transportation mechanism, where we talk of passive, active, and bulk, as well as factors limiting drug passage through the membrane. In lecture 5, which is receptors and its distribution, we defined receptors, receptor types, receptor drug or drug receptor interaction, where we mention affinity and efficacy, then drug distribution, factors affecting drug distribution vis-a-vis protein binding and what have you. Then we mention relationship to drug action and side effects. Now, to conclude this lecture, I have provided a basic knowledge for you on how drug interacts with the body, both at molecular and cellular levels, focusing on their movement across membranes, interaction with receptors, and distribution throughout the body. Finally, there are 3 review questions from topic 4 and topic 5 respectively for you to review your understanding of the topic. This is for topic 5. Thank you and God bless. Have a nice day.